Hi, it's Ms. Parrott, and this video is the first in a series about the central dogma of biology. The central dogma of biology is a principle that talks about how DNA, our genetic information, flows through biological systems. So that may be from one cell to the next, or from parents to their offspring. The central dogma of biology has two major tenets. The first is that DNA replication is semi-conservative, meaning that if you have an old strand of DNA, new DNA comes from that original strand. If you want more information on that, check out the video on DNA replication. The second major tenet of the central dogma of biology is that DNA is transcribed into messenger RNA, which is then translated into a protein. Then that protein is going to do a job in its cell, and a phenotype is going to be expressed. We're going to focus in this video on this first part of the second tenet. So it's all about transcription, which is the first step in protein synthesis. So transcription takes place in the nucleus, and basically we're making a copy of DNA into messenger RNA. Now the reasons that we need to make a copy of DNA, one is that we need to keep the DNA safe in the nucleus, so the DNA never leaves the nucleus, and also because DNA is a huge molecule with all of our genes, and not all of our genes need to be expressed all the time, right? Like my hand-making genes are in my eyeballs, but they're not expressed. Those genes are turned off. So mRNA helps us make copies only of the genes that the cell needs at that time. It has really just two major steps. The first is that the enzyme RNA polymerase is going to come in, unwind and unzip our DNA double helix by breaking the hydrogen bonds between the nitrogenous bases. And then that same enzyme, RNA polymerase, will add complementary nitrogenous bases to one of the DNA strands. So you can see the DNA strand here, ACT-GAC, and then RNA polymerase will come in and add the complementary bases. Now remember that mRNA is a little different than DNA because it has uracil in place of thymine. And then finally, that mRNA strand is going to break away from the DNA and leave the nucleus, go into the cytoplasm, and reach the ribosome ready for translation. Those are the big principles of transcription. Check out the next video to learn more about translation.